ARS Bidar, US Raichur, and sir experience is regarding about in the research they have worked about 8 years and in extension about 17 years, and he has total about 25 years services, and he have teaching in diploma and PG teachers, and total they have technology have been released to the universities about 15, and national and state level projects have handled about 4 as a PI in NHM, IFS, JDBNF and chemical testings, and as a core PI as a 6 project have been handled, and total PG student is about 1 PhD is completed, and they have sir have been guided about 9 MSc students. This is the detailed brief biodata of the Dr. Sunil sir. Once again, I welcome to the Sunil sir for the this national farming guest lecture. Thank you once again.
Thank you, Dr. Raju. So, today my topic is, as you know, effective disease management strategy through non-chemical methods. Friends, it is very difficult to talk after lunch. So, friends, after lunch it is very difficult to talk because kindly cooperate with me, but it is a very important topic. As you know, for any successful of this natural farming and also organic farming, our disease and pests are coming in the way. As you mostly taken the feedback from earlier scientists, C.P. Chandrasekhar and Pandit Rathod, the main constraint in organic farming and natural farming is disease management and pest management. It is a very big hurdle for farmers. So, slides are very more, but the pictures are more. As you know, let me start some brief about introduction. As you know, Indian economy or agriculture contributes 19.90 GDP. And also our advanced agriculture, the 3 major achievements, as you know, increase in productivity and reducing rural poverty and meeting food security. In our after Green Revolution, as you know, productivity and poverty somewhat reduced, and also food security needs we have met. Just you see there is a 5-fold increase in the food production. As you know, 50 metric ton to 275. And one more important, our poverty of the nation, 60% to 40% reduction. And life expectancy got doubled, 34 to 68 years. However, human population also increasing year after year, and also you might have known our Indian population is mostly highest in the world. It leads to more pressure on land and other natural resources. Due to this, as you know, there is a soil health degradation and also depleting water table and expanding multi-nutrient deficiency, declining in productivity, and also lower and stagnant crop yield, Main is pollution. So as I'm dealing with the plant production, let me know the pesticide world scenario. With respect to countries, usage, gram per hectare. So Japan is highest, 10,000 gram per hectare, whereas our India, 450 gram per hectare. It is comparatively less, and African country, 127 gram per hectare pesticide usage. And also you can see the consumption pattern of the pesticide in India. So, Raju, Ananda, idu ada noಡ್ರಿ. So this insecticide consumption is more in India, but in world, fungicide consumption is more, percentage wise. Coming to crop wise consumption of the pesticide, cotton occupies a major pesticide consumption. Thank you. So 52% of pesticide insecticide consumption in India, more compared to fungicide. But in the world scenario, fungicide consumption is more compared to insecticide. So crop wise, cotton is pesticide consumption is 52%, whereas our staple food rice 18%. It is very threatening. And vegetable and fruits 7%, wheat 6.5%, so groundnut 2.5%, sugarcane 0.7%. So pesticide usage in India state-wise, so Maharashtra is major 13,243 metric tons. Next stood Uttar Pradesh, and our Karnataka 1930 metric tons. So as you all of you might know, this Punjab School of Public Health survey during 93 to 2003, you increased use of pesticide. So cancer patients are increasing day by day. As you know, there is every night 9:30 p.m. one train called cancer train from Batinda to Bikaner. Total 60 patients will try. They are traveling from Batinda to Bikaner, that is locally called as cancer train, due to our pesticide consumption. It is very alarming. You see some pictures, it affects the food and fruit quality, and also human health, cancers, and also groundwater it will affect, and also animals. But due to awareness, pesticide consumption also pattern is decreasing year after year. So from 1955, again in up to 1988, it is increasing trend. And in 2012-13, it is somewhat decreasing. In India, there is a totally 215 pesticide. Out of that, 39 have been banned, for your information. You see, in apple in England, they spray 16 times of pesticides with 36 different pesticide residue. Some doctors are telling you eat apple daily, you can avoid doctor, but it is scenario is different. Our mango export over the year, it is decreasing due to this pesticide residue. In 11, our volume of tons 63,000. It is in 14, 43,000 tons we have reduced due to pesticide residue. But when compared to chemical and biopesticide consumption in India, as I said earlier, slowly due to awareness it is coming down, and biopesticide usage is increasing year after year. So our disease Loss is ranges between 15 to 20%. Comparatively, horticulture crops are more prone to diseases. The loss is more in perishable and storage losses that lead to aflatoxin production and food spoilage. This is really concern over food safety and security in India. Looking to this all challenges faced to our agriculture sector, we have to reorient our agriculture production system. We have to shift in input intensive agriculture to sustainable agriculture. That is natural farming and organic farming are potential approaches. This is the two natural farming and organic farming. They are coming a very good option in this direction. So let me come to our main topic. So there is, I want to brief some that in disease management, our natural farming management strategy is very meager. So I chosen this topic as a non-chemical methods of disease management. It includes all the methods except chemical. Coming to cultural practices, it is one of the major non-chemical methods. As you know, summer deep flowing, it is a very important non-chemical practice. It is, as you know, soil-borne diseases are very difficult to manage. This is the best weapon to control. As all pathogen propagules exposed to this heat. And good agronomic practices and crop diversification that includes roguing and crop rotation. So that is also one of the best strategy. This is the some. Huh? I will cover next. What you want? My topic is non-chemical methods of disease management. Under this, natural farming will also come. It will come in next. What? Next slide, I will cover. Very meager in natural farming, I will tell. So this is summer flowing. Monitoring is one of the important aspect in management strategy. Roguing of the disease affected branches and plants, disease plant residue removal at the early stage. Use of resistant varieties like in tomato for bacterial wilt is a major menace. Arca aba, arca alok, PKM1, they are resistant to bacterial wilt and nematodes. Bendi, Arca anamica, yellowing mosaic disease is a major disease. So brinjal, Pusa purple castor, again it is a bacterial wilt resistant. Chili, as you know, Moda complex is a major disease for that Pusa jola. And chili, potato, blight disease, Kupri Jyoti, Kupri Pukraj and Kupri Naveen. Our horticulture university, they have developed Sakre Bale. It is highly resistant to Sigatoka leaf spot disease. It is highly resistant to Sigatoka, one of the major foliar disease. So again, one more method in cultural practice, sowing method that includes early sowing and raised bed. As you know, the importance of raised bed. Weed management, as you know, they act as a alternate host, also a collateral host for many diseases. The weed management through intercultivation and use of natural enemies and fast-growing intercrops. In case of removal of collateral host, Alternaria solani, early blight. In case of tomato, that collateral host like that Solanaceae family, they will harbor in that weeds. Alternate host like in case of blast of paddy, Panicum repens, that acts as a alternate host for we have to take care of that. Seed treatment with Bijamruta, Jivamruta, cow urine, ash, cow dung, neem oil, different plant extract powder that will take care of our seed-borne and soil-borne disease. These are the some pictorial. Potato, Sclerotium rosea, collar rot, by our trichoderma seed treatment, root dip with some our bioagent cultures, use of plant extract. That principle includes induced systemic resistance by synthesis of antimicrobial, antiviral substances, processes like repellent and antipedant power vectors in virus disease. Botanicals, as you know, they act as a poisonous plant and safe medicine. There are some I given in notes. The one are important like garlic extract, ginger extract, garlic extract. They are very effective against anthracnose disease, downy mildew, and bacterial disease. Ginger extract, they are very useful for powdery mildew, root rot, and leaf blight disease. And citronella grass extract, they are very useful for bacterial disease. Fungemia extract, it is effective against soil-borne disease. That methodology I have given how to prepare and how to method of usage. Neem seed kernel extract, you are using widely and farmers also using. They are very effective against fungal and viral disease. And pudina extract, they are also very effective against bacterial disease. Same plant extract, that Mimosa pudica, that is very effective against leaf spot and fruit rot disease. Oh, drumstick and leaf extract also having medicinal value against root rot and bacterial disease, also early blight and leaf spot. Marigold, as you know, marigold, it is a very effective against nematodes. So spraying of root and leaf extract of marigold, that will take care of our nematodes. And also growing at the base of perennial plants. Perennial plant in basin of the perennial plant. If you grow the marigold plant after incorporation, nematode management is very effective. And also wheat brown rust, root rot disease also take care. So sweet flag, bhaje powder, common term, that is very effective against seed-borne disease. It used as a seed dressing material, seed treatment. Tulsi extract at 1:10 dilution, fungal disease and nematode management. This is also used for nematode. There is two options for marigold plant and tulsi. So coming to biological control, so microorganisms used in biocontrol, you can look into this picture. So fungicides, they are major shares. 49 fungi, they are used for as a microorganism used in biocontrols. Bacteria, around 36 as a bioagents. So nematodes, some viruses, they are also used. So microorganisms usage pattern in biocontrol. As an insecticide, there are totally 50 bioagents they are used. Whereas fungicide, again 35. These are the major popular trichoderma and pseudomonas. So what is the main biological control mechanism? As you know, nutrition competition, site competition, and also antibiosis due to some antibodies production, and also induced systemic resistance like phytoalexins, etc. You are also very well acquainted with this, why these are giving importance, advantage. Quickly we'll look into this. It avoids environmental pollution, and it avoids the residual toxicity, adverse effect on beneficial, it avoids the adverse effect on beneficial microorganism. It is less expensive as compared to chemical. And avoid the development of resistant strain. That is important. As in case of pesticides, we will get resistance development. And also bioagent application is usually once and does not need repeated. But in contrary, chemical we have to use repeatedly. So bioagents are more effective, especially for soil-borne disease. It is very effective against soil-borne disease. As compared to chemical, there is no successful control. And also quantity used of bioagent is very less compared to chemical. And risk-free management with reference to phytotoxicity. You can see in case of chemical, there is a phytotoxicity if you use overdose and some again mixture with other chemicals. But in case of bioagent, very risk-free management. And at it, this biofungicide, it becomes part of modern large-scale agriculture and also help in increasing crop production. What are the success for biological control? Selection of virulent strain is very important because some farmers, they are giving feedback that biological control is not effective like that. This may be the reason. So strain should be local strain, effective strain. There should be sufficient growth and sporulation on mass media and advanced application. Always bioagents needs advanced application because it provides enough time to interaction between antagonistic and target pathogen. But unlike in chemical, so immediate effect is there. But in case of bioagent, you should apply advance. And also for success of this biological control depends on soil temperature, moisture, and should be optimum. And there should be sufficient organic matter. You might have known this food base, it is very important. And monitoring the population of the target pathogen antagonist time to time is important. What are the, once we applied the bioagent, what are the population of bioagent and target pathogen? Regularly we have to watch. And handling and production storage should be easy because it is very difficult. And it should be cheap and available easily. But we'll suggest so many bioagents, but availability is very question. So integration of biocontrol with tolerant resistant variety and chemical control may be more feasible. So integration of biocontrol with tolerant resistant variety, it is most feasible. So look into Pseudomonas fluorescens. So as you know, it is a PGPR, plant growth promotion bacteria, induced systemic resistance, biocontrol of many plant diseases. What is the main mode of action? This antibiotic production, you know, is several antibiotic like pyocinin, pyrrolidine, and oomycin A. What will this antibiotic role? This is a fungistatic and also inhibiting the spore germination and lysis of the fungal mycelia. Second important is heterophore production that will produce a ferribactin and a pseudobactin like siderophore, which reduces the supply of iron to the pathogens, thereby suppress their growth of the pathogens. And one more important mode of action is ISR, induced systemic resistance. There are several ways by which induced systemic resistance like production of phytoalexins and increased lignifications and also production of PR proteins, that is pathogenesis related proteins. So production of phytoalexins that act as an antimicrobial when pathogen come in interaction. So increased lignification, by this it is less accessible for cell wall degrading enzymes. So PR protein, like when that pathogen enters, that produces the PR proteins that acts as an antimicrobial, and again it is also a part of SAR, systemic acquired resistance. In case of cotton, this Rhizoctonia solani root rot, due to Pseudomonas fluorescens, when you used, it will hinder the availability of iron to the Rhizoctonia solani. By that, it will control the pathogen. In case of citrus canker, use of Pseudomonas fluorescens at the rate of 4 gram per liter, it reduces the citrus canker lesions. In case of important septorial spot, in case of flower, in case of flowering plant, chrysanthemum, bacterial bite of anthurium, use of Pseudomonas, we can control these both septorial leaf spot and bacterial blight in case of anthurium. So management of wilt of pepper. So wilt in case of pepper, it is a very important disease, in especially in I think in Malnad area. So Phytophthora caused by. In this, it includes Trichoderma harjanum, 50 gram soil application per vine, along with half kg neem cake during June and August, 2 times. And Pseudomonas fluorescens 1% drenching, 3-4 liters per vine during June and August, along with 2 sprays during June and August. That is controlling, giving very good control in case of wilt of pepper. So this is one more important disease in case of soft rot, bacterial soft rot. In case of crucifers, Pectobacterium curtorum, very good control given by Bacillus and Pseudomonas in case of this bacterial disease. So main more due to PGPR, more greenness and growth in plant spread with Pseudomonas and Bacillus. In case of this wine crop, this Mallikarjun, ಯಾವ್ದ್ರಿ ಇದು? Beans. So fourth mode of action is competition. As you know, it is a fast growing Pseudomonas fluorescens. Almost all bioagents, they are fast growing, competing with food and place. One more production of hydrogen cyanide production, that is a volatile inhibitor, check the growth of pathogen. That is a plant growth promotion antibiotic production like auxins and gibberellins, thereby and also by phosphate solubilization. These are acts as a hormones, phytohormones. What are the method of use in case of Pseudomonas fluorescens? Used as a seed treatment, 5 to 10 gram per kg, and it's also used as a seedling root dip, 10 gram per liter for 30 minutes. Soil application, 2 kg Pseudomonas fluorescens mixed with 50 kg FYM. That is the soil application method. In case of plantation crop, 10 to 50 gram Pseudomonas fluorescens per plant, along with 3 to 5 kg of FYM. In nursery bed, after in nursery sowing, half kg Pseudomonas fluorescens mixed with 50 liter of water, we have to spray. That will avoid the damping of disease. As a foliar disease management, we can use 10 gram per liter of per water this Pseudomonas fluorescens. There is a very good option. It can be used as a seed treatment, soil application. In case of plantation crop, individual vine we can apply, and nursery bed we can spray. As a foliar disease management, we can spray 10 gram per liter of water. Very good. And another one very good bioagent, Bacillus subtilis. It is also plant growth promoting rhizobacteria. And it produces many secondary metabolites, hormones, cell wall degrading enzymes. You see, this is acts as a very good biofungicide and bactericide, Bacillus subtilis. It controls both soil and airborne disease in case of vegetables. And it also used as a foliar application to control leaf disease in many crops. And this compete with, there is a mechanism, competition also there, PGPR is there, and and PR proteins also produces. Bacillus subtilis inhibit the plant pathogen spore germination and also disrupt this germ tube growth and interferes with the attachment of the pathogen to the plant. These are the mechanism involved in Bacillus subtilis. There is a lipopeptide activity that gives the disease control. The lipopeptide activity is there in case of Bacillus subtilis. The method of use, again, seed treatment you can use, seedling root dip also you can use, soil application at the rate of 4-5 kg mixed in 200 kg per acre, foliar spray 5 gram per liter. That is the method of use. So in case of that capsicum, grey mold, Botrytis cinerea, fruit dripping in Bacillus subtilis, that gives good control, Bacillus subtilis. So powdery mildew in case of pumpkin, Ericipe, that is managed by, very good management by Bacillus subtilis strain QSTS 713. So Sigatoka Mycospirilla piginensis, that is controlled by Bacillus subtilis strain QST 713, very good control. You can observe Bacillus subtilis 2 liters that gives visible strikes on leaves 3.9 when compared to second treatment Bacillus subtilis plus mancozeb. They are on par. So suppression of cucumber beetle, corn rootworm and also bacterial wilt by this Bacillus subtilis. One more important thing, Bacillus subtilis due to PGPR, that reduces the bitter cucurbitacins. Then this bitterness is there, though, that will avoid by this PGPR activity due to Bacillus subtilis, and also this reduces the bacterial wilt. This is also reduces the bacterial wilt. Again, one more popular biofungicide, Trichoderma. This is most popular biofungicide among farmers, that reduces the so many soil-borne pathogens like Sclerotium, Rhizoctenia, Pythium, Phytophthora, Fusarium, Macrophomina, that help in reducing bacteria and nematode population also. Major effective Trichoderma species, as you know, Viridae, Trichoderma viridae, Argonium, Hematum, Coningi, Viriens. So look into its mode of action. As you know, Trichoderma also produces so many antibiotics, namely dermin, viridin, gliotoxin, trichodermin, acetaldehyde, which will help in reducing seed and soil-borne pathogens, antibodies. It also induced systemic resistance and competition, mycoparasitism. These are the major mode of actions. Again, method of use, it also used as a seed treatment, soil application, also plantation crop, individual plant we can apply, and also foliar disease management at the rate of 10 gram per liter. That is a very good control. This is one pictorial mycoparasitism in case of Trichoderma species. So this is the penetration and Hastoria formation within the hyphae of Rhizoctonia. That Trichoderma, it will penetrate in case of Rhizoctonia solani and also Hastoria formation within the Rhizoctonia. And there is also that penetrant host cell wall by severe secreting lytic enzyme, that will penetrate by secreting the lytic enzyme, and also there is a coiling mycoparasitism in case of this Trichoderma. Antibiosis, another mechanism, antimicrobial compounds by antagonistic fungi, that is glyovirin, that is antibiotic produced. You can observe glyovirin non-amended media. This is a glyovirin amended media. In glyovirin amended media, there is restrict the growth of that Rhizoctonias solani. Very clear-cut demarcation. This is the enrichment of FOM trichoderma application. In case of nursery, also trichoderma added in potting mixture. In case of strawberry, botitis, that fruit rot, it is a very important disease that is giving good control by this trichoderma. Even you can give in drip irrigation and also root dipping you can do. This is the one most important against nematode management, Bacillomyces liliaceus, that parasitize the nematode eggs, and also it is proved to be effective biocontrol of nematode disease. It is used as a seed treatment 4 to 5 gram per kg. Again seedling root dip at the 10 gram per liter of water for 30 minutes. Soil application 5 kg Bacillomyces mixed in 200 kg of FYM for 1 acre. For again plantation crop, individual plantation crop, so 10 gram per plant mixed in 3 to 5 kg of this FOM. This is a nematode management due by this Bacillomyces. That is Melocom, that is trade name of that one bioagent that is giving good control of this nematode in case of this citrus field. So in case of Phytophthora arecae, choleroga, very important disease. One of the non-chemical method is first one is spraying of Bodo mixture. You can see the fourth treatment, covering the bunches with poly covers before monsoon. That is very effective in case of disease incidence reduction, and also you can see the net profit 1 lakh as against this spraying Bodo mixture and covering the bunches with polyols. Fourth treatment is giving very good management. That is covering the bunches with poly covers before monsoon. You see, that is a very good practice in Malnadi area, covering the bunches with poly covers before monsoon. One more, management of drainage channels in areca nut gardens, that is giving good control in case of areca nut disease. Severity of leaf spot areca nut in nursery by use of this bioagents. Pseudomonas fluorescens in case of leaf spot management in areca nut. That is giving a good control and also with net return as compared to chemical treatment. In case of fifth treatment, Pseudomonas fluorescens at the rate of 0.5%, you see the PDI and also net return. It is the best option, Pseudomonas fluorescens, in case of management of leaf spot. In case of coconut bud rot, it is an important disease, that treatment 5 gram per each crown, that is Pseudomonas fluorescens and Trichoderma viridae, that giving very good control as compared to other treatment, very good control. Black pepper, that foot rot and quick wilt, one more important disease in case of black pepper, that is controlled by soil application FIM 20 kg and neem cake 1 kg per vine, and Trichoderma harjanum native strain 50 gram per vine, and mulching with plastic sheet 200 micron, that giving the good control in case of this black pepper put rot. As you know, use of cakes like neem cake and pangemia cake and other cake, 250 kg per acre, which will enhance the antagonist microbial activity, thereby reduces the soil-borne disease and also nematode infestation. Intercrop and trap crop, cover crop. The marigold, 2 lines of marigold, intercrop at every 10 lines of vegetable that manage the nematode disease by producing alpha terphenyl, that is allelopathic effect on this nematode species. And also marigold acts as a poor host for nematode. And one more, growing up 1 line of tomato as an intercrop with 10 lines of brinjal. That reduces the nematode infestation as well as other soil-borne disease. 1 line of tomato at every 10 lines of brinjal, that reduces the nematode infestation and also some soil-borne disease of brinjal. This is the photograph, marigold intercropping. Coming to indigenous technology knowledge, like natural farming practices. First one is Bijamruta. As you know, in case of natural farming, it is a very popular usage. Bijamruta enhances the germination ability and also reduces the seed-borne disease. In this, important ingredients are desi cow dung, lime, desi cow urine. Method of preparation is given in your manual. So it is used as a seed treatment which enhances the germination ability, also reduces this seed-borne disease. Shunti astra, that is used as a fungicide to manage almost all fungal disease in natural farming. So major ingredients are desi cow milk, dry ginger powder. There is a protocol, boiling desi cow milk and a lot. You can I tell this protocol? Not necessary, no? Already covered. It is very good fungicide. Yes, it is very good fungicide against almost all fungal disease. So it has given like anthracnose, powdery mildew, leaf spot, rust disease, amele purple seed stain. Very effective against this disease. And one more, instead of shunti, you can use pepper powder. Again the protocol same, desi cow milk, instead of dry ginger powder, you can use pepper powder also. One more fungicide. And one more fungicide like buttermilk. So 5 liter buttermilk in 200 liter. So the important is 5 days buttermilk is very important. And that too, you can, if you use 40 days old buttermilk, that is most effective against all type of plant disease. They are telling all type of plant disease, but we have to study in detail. All type plant disease means it includes fungal, bacterial. So, but in our study, in our project, it is effective, shown effective against budi roga, powdery mildew, anthracnose, rust, and also leaf spot. It is effective against. One more fungicide, kakulli extract. Simple preparation, 5 kg desi cow dry dung, means kulla, dry dung, cow dung, mixed in 200 liter of water. You have to keep for 24 to 48 hours. Mix thoroughly 3 times every day. That is important. It is also acts as a very good fungicide. And Jivamruta, it acts as a double advantage. Plant growth hormones as well as manage fungal and also bacterial disease. Very good advantage. His method of preparation is very simple. Add 20 liter of cow urine, 2 kg cow dung, 200 liter of water, 48 hours incubation, mix thoroughly 3 times every day. Very effective against our rust disease, powdery mildew, bacterial yeast spot. It is very effective. So it also acts as a hormone. Sometime it will help us. Neemastra, in case of viral disease, for control of vectors. So used for manage the vectors responsible for viral disease. So you can major ingredient are desi cow dung, cow urine, neem leaves, fresh neem leaves. In case of this sterility mosaic disease, in case of red gram. So one more yellow mosaic virus disease in case of green gram, that controls vector like white fly and also mite, Neemastra. Neem paste like gummosis disease caused by Phytophthora. This is a very good answer for this gummosis type disease. We have to scrape those affected portion. After this, you just paste this neem paste after preparation. Panchagavya. It is used as a plant hormone again, just like Jivamrita. And it supply the new micronutrients. And also major mechanism is it increases the disease resistance and used for powdery mildew, downy mildew, leaf spot. It increases the disease resistance, supply the micronutrient, but method of preparation is somewhat tedious. It requires so many ingredients, cow dung, cow ghee. Our farmers are not having patience to prepare this. So spraying Panchagavya at the dose is 3 to 10% based on the crop. At flowering, we have to spray 2 times at the 15 days interval. So coming to viral disease management. So as you know, viral diseases are very difficult to manage. Nowadays, viral diseases are making lot of losses to the farming community. As you know, there is no effective viricide. What is the ways in case of organic and non-chemical methods? Just to have a look into this viral disease management. ಹೊರಡಾಗ್ತಾ ಇಲ್ಲ. ಸ್ಟಕ್ ಆಯ್ತಾ? Why organic? It is answer to all disease, not only viral disease. It restores our natural resources and pesticide residue free commodities, safeguard to our environment, and also control pest and disease without harming our environment. So what is the eco-friendly management in case of viral disease management? Use of virus-free propagules of crop plants, phytosanitation, cultural practices, control of vectors, cross protection. In first use of virus-free crop plants like seed and planting materials from certified source, seed treatment with biological agents and also plant extract, meristem tip culture technique. So by virus-free planting materials we can produce. Nursery bed covered with, as you know, in case of virus disease, that vector early stages we can avoid by use of nylon net 40 mesh size. After that we can healthy seedling raised from nursery protection. So this is very good. Next one is sanitation at early stage. We can avoid the further development of virus disease. So So at early stage, we have to remove the diseased branches and plant residue removed and burned, removal of voluntary plants, alternate host, and also eradication of overwintering host. These are some example. Weeds are important source of infection, as in case of rice tungro, echino chola, crus gully, and bud necrosis in case of groundnut, that is tegatus species, sunflower. As you know, this parthenium also act as a reservoir for virus, and green gum, yellow mosaic, Acalypha indica, that is reservoir acts as a this weed for this virus. Soil solarization is a very important management strategy in case of virus disease management. So it raises the temperature 45 to 50 degrees centigrade for 10 to 15 centimeter top, and after 15 to 30 centimeter depth, again temperature raises to 40 to 45 degrees centigrade. By this we can control some viral disease. Cultural practices like plant density, crop planting date, and barrier cropping, intercropping, and trap cropping. These are the important management strategy in case of cultural practices. First one is alteration in sowing date. So almost avoiding the exposing young plant to infection. That is an important strategy in case of management. So we have to, in case of maximum vegetative growth should not coincide with the period of maximum hectare activity. So during vegetative growth, it should not coincide with our maximum hectare activity. That is the most vulnerable stage. By that, we have to alter the sowing date. Some examples in case of groundnut, there are so many examples. Let me tell groundnut peanut bud necrosis, early sowing in Kharif, sowing in June rather than July, where that population are very lower in case of June. That peanut bud necrosis virus is thrips. The activity of thrips very less in case of June sowing. And one more example, potato virus sowing between October to December, that is avoiding the less vector activity during vegetative growth stage of the potato. This is one more effect of barrier cropping. In case of, for example, viral disease, if you sow the barrier crops like maize, pearl millet, that reduces the viriparous apid movement in case of some example like stylet-borne viruses, like green gum mosaic virus, and other in tomato, leaf curl virus. So after entering in barrier crop, that will lose the infectivity, that vector. After passing that barrier crop, it will enter as a virus-free apid. That is the main mechanism. Example in case of green gum, groundnut bud necrosis, sowing the 4 lines of pearl millet before sowing up the green gum. We have to sow the 4 lines of pearl millet. That will avoid the groundnut bud necrosis virus movement, that like thrips, and also that okra, yellow vein mosaic virus, where the barrier crop like maize, that reduces the hopper movement. That is a very good technology. And effect of intercropping, that is also one more important non-chemical methods. The use of non-host crops in interplanting can significantly reduce the rate of virus spread. The intercropping should be non-host for that virus. For example, one study is there that cassava intercropping with pepper, plantain crop intercropping with pepper, that reduces the viral disease severity to 4 and 7, and also viral disease incidence also reduced. That percent disease incidence and severity of this PVMV on pepper vein mosaic virus on pepper, when you intercropped with cassava and plantain, that reduces that PVMV virus infection, because those are non-host crop. This is also some study of intercropping, groundnut plus bajra, very good intercropping system that reduces the groundnut peanut bud necrosis viruses. One more vector control by altering vector efficiency or behavior by interfering with virus multiplication, just like resistant variety. By developing resistant variety, we can interfere with virus multiplication, and by using botanicals. Second one is interfering with vector behavior, just like use of traps, mulches, we can interfere with vector behavior. One more, oil spray that interfere with transmission process. Just we'll see in detail. You can see the one more is resistant cultivars. Some resistant cultivars can also use it as a very good method in case of management. Some rice tungro disease, there are some cultivars like Barhani, IR36, groundnut peanut stem necrosis, Kadri 3, Kadri 4, red gram sterility mosaic, Asha, and Bendi, yellow vein mosaic, Arca anamica, and tomato leaf curl virus, Nandi, Sankranti, Vaibhav, Arca Rakshak. So, botanicals, by use of botanicals also, the main mechanism is induced systemic ISR by synthesis of antiviral substances, and also it acts as a repellent and antipedant. Some important botanicals we have listed here: neem leaf extract, garlic clove extract, bougainvillea, gliricidia, sorghum, clerodendron, parthenium. There is some study, effective gliricidia leaf extract, sorghum leaf extract, clerodendron leaf extract. There is a very good management of tomato leaf curl virus, and also reduces the whitefly vector population. This interfering with vector behavior, as I told, use of mulches, yellow sticky top, that aphid respond differently to various wavelength light. The use of attractive colors traps, any traps or repellent to avoid the landing of the vector on susceptible load. It may trap or repellent this different colors traps. You can use the mulch like plastic mulch, aluminum mulch, sawdust, straw husk mulches. The use that you see this photograph, plastic mulching that reduces the infection, that aphid landing. The mulches used in between the crop reflect the sun's ultraviolet rays and mask the light emitted by plant. That is the main mechanism. The mulches used, that plastic mulches between the crop, reflect the sun ultraviolet rays. And aphid, in the absence of signal, keep flying, which enable indirect disease control. And yellow sticky trap for control of greenhouse vector, nylon net to avoid the physical contact between insect and plants. There are some study, polythene sheet, aluminum foil, that reduces the aphid population. Oil spray, as in case of papaya ring spot virus, one of the management strategy, we are using vegetable oil at the percent of 1%. So the main mechanism is interfere with insect ability to acquire and transfer the virus particle. One more is it block the air holes. By that it will insect breathing it obstruct. And also may disturb in feeding, and also non-polluting fit well in that IPM strategy. There is no resistance development in case of oil spray. Organic amendments also using in case of virus disease management. That mixture help in proper growth and functioning of plant, resulting in active metabolism of the plant, leading to reduced disease incidence of maximum control. And organic oil like neem oil, effective in limiting the hatchability and adult emergence, adult white population. Just like spent effect of organic nutrition amendment in case of cotton leaf curl virus. One study is there, spent compost plus leaf compost, and one is kitchen waste plus leaf compost, that giving good control, less disease incidence in case of spent compost plus leaf compost. One more principle is antiviral principle, extracted from fresh leaves on non-host sunflower necrosis virus in case of sunflower. Prosopis plus goat milk, that reduces the percent infection in case of this sunflower necrosis virus. And again second treatment Bougainvillea and plus goat milk. Antiviral principles, the main mechanism is the reduction in case of virus infection is due to different kinds of action of AVPs on either virus or host plant. The antiviral principle induces the physiological changes in the host which account for increased resistance. And third one is the treated plant with increased level of total phenols like PAL activity, PPO activity, that is polyphenol oxidase, phenol oxidase. AVP act as an inducer of plant defense mechanism. These are the major AVP mechanism. These are the study. One more, cross protection in case of virus disease management, a very good example in case of citrus testisa and papaya ring spot virus. The major mild use of mild or attenuated virus strain to protect the against economic loss by severe strain. Very good technique used in case of this papaya ring spot virus and also citrus testisa. There are some examples given in case of cross protection as in papaya and also in citrus. Biocontrol agents like predator, parasites, entomopathogens for a vector control. Some examples I would like to tell. This red gum dry root rot caused by Macropomena fezolina. It is a really nightmare to our pulse ball farmers. Nowadays, during last 2 years, there is a severe outbreak of this disease. Since this disease is very difficult to manage, it is seed-borne, soil-borne, and airborne. The major symptoms like root rot, it at flowering and podding stage, it start appearing, and lateral roots are damaged, just like some spindle-shaped symptoms. Xylem phloems are blackening. Last year, there is a total 1 lakh hectare damage due to this disease. Very severe disease this. We'll look into the what are the options in case of non-chemical methods in this disease. You see the picture of this severely affected plot of this red gram. Whole plot is affected by dry root rot. What are the options in case of this non-chemical methods in this dry root rot? So, as it is a seed-borne, soil-borne, air-borne, the options are left with subbed deflowing is important, crop rotation, mixed or intercropping with sorghum, seed treatment with trichoderma 4 gram per kg, soil application 1 kg trichoderma enrichment plus 100 kg powdered FOM plus 20 kg neem powder. That is very good effective against this Rhizoctenia bataticola. And rogging of infected plant at initial stage. 1 acre for 1 acre. And one more last irrigate the crop at pre-flowering stage. That really works better. Since the epidemiological factor for this disease aggravate when the temperature rises above 28 degree coupled with moisture stress. So, if you give one light irrigation at the time of pre-flowering, that reduces the disease incidence. We have seen several plots, but most of the farmers are not having that facility. Wherever it is there, you give one light irrigation. And one more important disease that is a national disease like pomegranate bacterial leaf blight and wilt management in case of pomegranate. There are some protocol is there. In case non-chemical method, foliar spray of cow urine, buttermilk, and also for wilt management, foliar spray drenching is there, and some trichoderma application per tree per year is there, per year. Foliar spray of Pseudomonas is there. Soil application of boron, zinc. Total package for bacterial leaf blight and wilt management. You see the some severely affected bacterial blight. Especially that 10 gram per liter at 15 days interval, this Pseudomonas fluorescens, it is working very well, especially for this bacterial leaf blight. Spraying of Pseudomonas fluorescens at the rate of 10 gram per liter at 15 days interval, and also paste on infected parts after pruning, and soil application before planting 10 gram per plant with Pseudomonas, that giving a very good management for this bacterial blight. Again, ginger rhizome rot, it is a very important disease in case of ginger. Good agriculture practices that includes land preparation, date of sowing, and selection of the land, everything. One more key factor is selection of healthy seed material is a very key important factor. Storage of ginger rhizome in plastic sheet at 47 degree centigrade for 30 minutes. That kills the pathogen population in case in the seed material. Seed treatment with Trichoderma Pseudomonas at 1% for 30 minutes. Again, rogging at an initial stage. Soil solarization. This is the ginger rhizome rot incidence. And rabbing one more, burning the prepared seed bed before sowing with paddy husk. It is one more practice for overcome the soil-borne disease. Rabbing, it called as rabbing, burning up seed bed before sowing with paddy husk, dry leaf material of sugarcane. Per square meter 6 kg we can burn. Chili Muda complex, you can, it is a complex disease. So, neem cape soil application, seed treatment, rogging, spraying with NSKE, Sorghum leaf extract, again an antiviral principle. So foliar spray with vegetable sulfur, it is advisable in case of organic. This is grape powdery mildew, anthracnose. In our project study, 4-year study, we are having this natural farming project. We have studied in case of black gram, powdery mildew. So 4 modules: natural farming, organic farming, POP, and farmers practice. In this farmers practice, giving good management, followed by just organic farming, that gives the good control. ಆಯ್ತು ಇಲ್ಲ ಇನ್ನೊಂದು 4 ಸ್ಲೈಡ್ ಅಷ್ಟೇ, 4-5 slides. So package of part is giving good result. This is the photographs, different treatments. So in case of Bengal gram, that is soil-borne wilt disease, again our package of part is giving good result. So followed by organic farming. In case of soybean pod blight, that is caused by Cercospora, and in this also package of part is giving over the 4 years, giving good result, followed by organic practice, and third one is natural farming. In case of Cercospora leaf spot, in case of radgum, same trend. In case of dry root rot of red gram, the package of practice giving good control, followed by organic farming, and third one is natural farming. Coming to merits of this non-chemical methods or organic methods or natural farming, merits means this healthy soil supports the robust plant growth, that resist for the disease. Biodiversity helps in maintaining the balanced ecosystem. Reduction of external input. ಆಮೇಲೆ one more long-term efficacy. That is the important. Low cost effective as they involve readily available natural substance. Demerits are, it is time consuming. One more is knowledge intensive, because deep understanding of ecological process may involve experimentation, complexity is there. Cost because yield variability is there during transition period. So yield may take, that consistent high yield may take time. Less disease control. In our study of project, less disease control in some organic methods may not be effective in controlling the diseases, leading to potential yield loss. Variable efficacy, limited spectrum, may not provide broad spectrum disease control. It won't control the broad spectrum more than one disease. So conclusion, non-chemical methods offer sustainable and environmentally friendly solution for plant disease management. Each method has its merit, demerits, but their effectiveness can be maximized through integration of multiple methods. We can use best practices in natural farming, organic farming, and ITKs. We have to integrate in this scenario. The dose and ingredients used in this preparation of different natural farming formulation in natural farming are to be standardized scientifically for efficient control of pest and disease in different crops. Still, we have to do more experimentation. The future of plant disease management lies in the balance between productivity and ecological sustainability. It is between productivity and ecological sustainability. Thank you. Madam, you are asking some? Organic farming. That is why the conclusion I left with this integration of all good practices. So I selected non-chemical. Whatever the non-chemical options, it includes everything: cultural practice, botanicals, organic, natural. Yeah, not exactly natural farming.
Okay, if anybody have any doubts, you can ask to the experts. Otherwise, we are going to answer the sir. Today, sir has explained regarding about detail the pest and disease management in the natural farming as well as in organic farming, and they have conducted the 4 experiments in Zone 1. They have explained regarding the all things and management in the organic farming by using the cultural, physical, mechanical, and some of the the microbial agents. And in case of natural farming, also they have recommended Bijamrita, Jivamrita in details, which are the pest and disease can be managed in the natural farming by different use of different methods. In this content, in the behalf of all the participants as well as in the name of US, this one, University of Agriculture and Horticulture Sciences, Shivamogga, as well as all the participants, behalf of, I am going to thanks to the Dr. Sunil sir, one and all. Thank you.